This video, we will discuss how to use a dichotomous key to help identify bacteria. When using a dichotomous key, you're looking at the colonial characteristics of the bacteria. You have a copy of this dichotomous key in your lab manual. We grew 4 different species of bacteria in petri dishes for you to identify. We have Bacillus subtilis, Sarcina aurantica, Enterobacter coli, or E. coli as you would know it, and Serratia marcescens. Now, to use the dichotomous key, you always start at the top and work your way down. So let's start at A. If we go to A and go to number 1, it says colony size large, and does it have a rough edge? So we would look at our bacteria in these dishes very closely and see, does that colony look large and does it have a rough edge? Now, I know large is a very vague term, but if you're looking at these 4 different dishes, you're looking at the small circles to try to see what bacteria is growing in there and about what is the size of those. Now we have these over here, which are actually pretty small colonies. All right. And then we have these here, which are these larger circles. We would consider that a larger colony. The other thing you were looking for is, does it have a rough edge? If we look closely at these tiny little colonies, and I know it's kind of difficult to see in the video, but you're looking at these individual colonies. So do they have a perfectly round edge, or is that edge kind of jagged looking? If you look at this colony next to my finger right here, you can see that it has a little point on it. Right? So it's not a perfectly round colony. You can also look at the edges over here that has a very rough edge to it down here towards the bottom of the petri dish. So if the colony is large and it has a rough edge, then we know that it's Bacillus subtilis. If the colony is not large and does not have a rough edge, then we go to number 2. Number 2 says, is the colony small and does it have a smooth edge? If the colony is small and has a smooth edge, it says go to B. If we go to B and go to number 1, it says, is the colony opaque? Opaque means that you cannot see through it. Right? If it is opaque, then we know it's Sarcina lutea. Now, one thing I want to point out is that we have Sarcina lutea on the key that you're using, but we actually grew Sarcina aurantica. They look very similar, so we can still use this key for identification purposes. Now, when you're looking at Sarcina, and we'll focus on these small circles up here by my finger, you can see that they're opaque, meaning you can't see through them. They're solid colonies, right? You can't see through them, right? So again, if it's opaque, then we know that it's Sarcina. If it is not opaque, we go to number 2. Number 2 says, is it transparent, or can you kind of see through it? Does it have a very glossy, clear look to the colony? If so, it says go to C. Letter C, we'll go to number 1. Number 1 says, is the colony cream? If it is cream, then we know it's E. coli, which we can see here. As you can see, E. coli is translucent, meaning you can see through it. So if you look at my fingers, you can really see through those colonies. And it has a cream color. So again, it's transparent. It has a cream color, so we know it's E. coli. Now, if it is transparent and it does not have a cream color, then we'll go to number 2. It says, is it red? We can look at Serratia marcescens and see, all right, yes, it's translucent or transparent. We can kind of see through these colonies. And they have a red color. So for your practical, you'll need to be able to use this key. What we will do is we will pick one of these 4 bacterial species. We'll erase the name on these dishes. We'll put one out there on your practical question and have you identify the bacteria using this key. Again, these names will not be written on there. If you have any specific questions on how to use the key, please let your lab instructor know.